Whenever a wrong transfusion of the blood takes place, it is going to cause the so-called process of hemolysis. That means the blood is going to damage. To have a correct understanding of that particular blood transfusion from a blood group to a specific blood group only, we must know about the inheritance of this particular blood groups, which can be expressed by the term multiple alleles, and these multiple alleles are the causative factors for that particular inheritance. Generally, the child receives one of the haploid set of the chromosome from both the parents. That means they are going to inherit only one allele from that particular parent, and these alleles they segregate and they combine, and finally they will have the expression of the gene. And to understand that, let us study the multiple alleles, how the blood groups are designated as B, AB and O and AB. For this, let us begin with ABO inheritance system with multiple alleles as an example. The topic is on multiple alleles. With reference to inheritance in human beings. Generally, a gene has two alternative forms or versions. Now, they are present on a homologous chromosome, a pair of homologous chromosome at the same locus. Locus means the position on the chromosome. The plural is loci. This is the homologous pair. Now, a gene called a dominant gene A, and this is the one which is being present at the same locus. I told you that it has two alternative versions, and the other alternative is this. And there is a situation where we call it as a dominant and a recessive. What is dominant? The one which can express even in a single dose is called as a dominant. The one which can express only in double dose, it is called as a recessive. Now the two alternatives of the gene is either dominant or recessive. They are present on the same locus of the chromosome. Now altogether there are two versions of the genes, two forms of the gene, which has given three genotypes. What is a genotype? A genotype is none other than the genetic constitution of the organism. This is the gene, that is the one which is being present on the chromosome. That is what is called as the genotype. Sometimes there will be more than two forms of the gene. As in biology, we call it as more than two, it is called as multiple. They are the alleles, more than two. If more than two are present, they are called as multiple alleles. Now, as I told you that the genotype has only two types, two alternative forms. But in population of that particular organism of a single species, specific species, it is the one which is being more than two, it is called as multiple alleles. The existence of more than two allelic forms in a population, it is called as multiple alleles. Genotypes are present in individual or diploid organism, whereas multiple alleles are present in the population of a species. Now, how many types of genotypes are being formed with reference to the number of alleles? It is being given by a formula or by the expression n multiplied by n plus 1 divided by 2. The n refers to the number of alleles. Now the best example with reference to the multiple alleles is expressed in human beings, the so-called ABO system with 3 multiple alleles. Now the 3 multiple alleles means the n is 3, then when once we substitute this with reference to the formula, then it is going to be 12 by 2. There are 6 genotypes. Altogether, with reference to the blood groups, there are 6 genotypes in the ABO system. ABO blood group system with reference to the human beings. First, Landsteiner. In the year 1900, he has identified the blood groups ABO. Later, there is the fourth blood group AB, that is the one which is being expressed by 2 scientists by name Sturley and De Castro. Both of them are the students of Landsteiner only. Altogether, there are 4 types of blood groups with reference to the human beings. Now, this is what is being referred as the phenotype. Now, phenotypes of the blood groups within human population expressed by the blood groups as multiple alleles, they are in the form of 4 types of phenotypes: A, B, O and AB. Now, actually, according to one statistics, this is the one which is being referred as 47% of the human population is of O type of blood. The one which is only 3% is none other than AB group. Now, A group constitutes 42% and B group constitutes 80%. Now, altogether, the population of the human beings is predominated by O groups. It is followed by A group. It is followed by B group. It is followed, the list is none other than AB group. How these particular phenotypes of O, A, B and AB are being expressed? It is due to the presence or absence of either A or B antigen. This antigen is nothing but the one which is genetically produced. That is none other than produced by a gene. It has a name which is called as agglutinogen. It is also referred as isohemoagglutinogen. Now, this is the one which is being referred as the antigen. Either it may be A or B. That is the one which is being actually produced by the gene. And this is what with reference to the Landsteiner and his students have discovered. Now, according to this particular expression, now if it is having the antigen A, the blood group is A. If it is having the antigen B, it is the blood group B. If it is having both A and B antigen, it is being referred as AB. If it is not having neither A nor B, it is referred as O group. All these are the actual 4 phenotypes, and these 4 phenotypes are being expressed with reference to 6 genotypes. The presence of antigen A, constitutes the A type of blood, and that may have this genotype, or it may also have this genotype. Similarly, for the B group, it may have this genotype, or this may have this genotype. The AB group is having the genotype A and B. The O group is having the genotype this, and this may also be represented as this. So now all together 1, 2, 3, 4, 5, 6. 6 genotypes are there with reference to the blood group A, and with reference to the blood group AB, and with reference to the blood group O, which I call it as agglutinogen. The agglutinogen is none other than the one which is going to have an action with reference to the specific antibody. Now, this agglutination reaction is very specific in these particular blood groups. Now, actually, this is the one which is being present on the surface of RBC. Now, the antigen A, antigen B, and antigen AB, they are the ones which are being present on the surface of RBCs. Now, you see the shape of the antigen with reference to the blood group A. Now, this is the one which is I referred as isohemoagglutination. This is with reference to the blood group A. Now, diagrammatically, I represent the antigen B like this. Now you see the difference between A antigen and B antigen. Now in this AB group, now it has both. It has both AB, A antigen, as well as B antigen. Now that is what is being referred as AB group. In A group, they have neither antigen A nor B. But in terms of population, this is more. I already told you that it is none other than the one which is having nearly 47% of the population, O group. Now actually these antigens are polymers of sugars, sugar polymers. They are the ones which are being added with protein as well as with reference to the carbohydrate molecules. Now they are added with the carbohydrate and they are going to spin over the surface of the RBC, a blood component. Now this spinning results with reference to that particular antigen, and that is very specific in its shape. A has this, B has this, AB has both, and O has neither A nor B. Now this particular protein, I write four types of glucose molecules. Glucose number 1. The second one is N-acetyl glucose amine. Third one is galactose. Fourth one is galactose acetyl amine. The fifth one is none other than fucose. Now, for any type of blood, that particular O group, I write this as 1, 3, 2, 3, and this is the one added with the fifth. Now altogether there are 5 sugars. 1 refers to glucose, 3 refers to galactose, 2 refers to N-acetyl glucosamine, 3 refers to again galactose. This is added with fucose. This is the one which is being present in O type of group. In A type of blood, it is having this particular basic background of the carbohydrate molecule, but it is added with fourth sugar, that is none other than N-acetyl galactosamine. That forms A group. Now you see the difference between the basic and the A group. Similarly, if it is the B group of blood, basic component is same, but it is not added with 4. That is as per the A group. It is added with again that particular galactose 3. If it is added with 3, then it is called as B group. If it is added with 4, then it is called as A group. In AB group, it has both A and B. These are the main sugars, hence they are called as glycoprotein. Protein is genetically constituted and glucose is the one which is being added, and all there are 5 different types of carbohydrates. Break. A scientist is the person who has expressed that the so-called blood groups A, B and AB and O, these are due to that particular expression of the gene which produces the protein, and this protein is synthesized with reference to a gene that is present on the ninth chromosome of the human beings. Actually, human beings are having 23 pairs of chromosomes, homologous pairs. 22 pairs, they are autosomes, and the 23rd pair is none other than XX for female and XY in male. They are referred as the so-called allosomes. Now, on that 22 pairs of autosomes, if you take the 9th chromosome, at a specific locus, there is a gene by name I. Now, this gene I, it is having either A or B. At the same locus, it may have this multiple allele, or it may have this multiple allele. Sometimes it have both A and B. That is why it has expression of A antigen, it has the expression of B antigen, and for this group it has the expression of only antigen B. For this group it has the expression of the antigen A only. Now on the 9th group, depending on the allele of the particular gene, the antigen is being produced, and as a result the antigen, the blood groups are designated as A or B or O or AB. This is the work of Bernstein. Now, once we talk about this particular antigens, there is a specific antibody for this particular antigen. What is antibody? Antibody is none other than the component that is being present in the blood plasma. What is plasma? Plasma is none other than the blood component which is without a protein by name fibrinogen. Fibrinogen is a clotting factor. The blood cannot clot if it is removed, that particular protein fibrinogen. Now, once we remove that fibrinogen, the blood component is called as plasma. The plasma contains antibodies. These antibodies, they are referred as agglutinins. The agglutinogen is the protein, agglutinin is the antibody. For A antigen, the antibody is B. For B antigen, the antibody is A, just opposite, anti. Now for O group, they have neither antigen A nor B, but they have both antibodies A and B. Now for AB, they have neither antibody A nor B. That means both the antibodies are absent. For O group, both the antibodies are present. And this is what is being referred as agglutinin. Agglutinogen and agglutinin is the one which is having that particular agglutination reaction. And as a result, when any incompatible blood transfusion takes place, these two are very important. If they doesn't match, I told you that agglutinogen refers to, it produces the antibodies. That antibodies, if they are not native that particular blood, now it is going to cause a phenomena, damage of the blood, which is being referred as hemolysis. That is what is being referred as hemolysis. The antigen must match with the antibody. Then only the individual can survive. Otherwise, it cannot survive. Break. Blood typing. How the blood group is assigned, whether the person is A group or B group. I told you that there is a specific antigen and antibody. That what they are, in other terms, it is referred as agglutinogen or agglutinin. Agglutinogen is antigen, agglutinin is antibody. Blood typing is the one wherein there is a mixing of blood, and in that mixing of blood they take these particular RBC cells and they mix with that particular anti-serum. What is anti-serum? I already told you that serum is the one which contains the antibodies. They will mix the antibodies with the RBC in blood typing. If you take an antigen of the RBC and mix with anti-A antibody, anti-serum A antibody, or you can type with reference to anti-serum B antibody. Anti-A antibodies, anti-serum A antibody has the so-called antibody B. Now, anti-serum B antibody will have the antibody A. I already told you that A type of blood must match with the antibody. So, A is going to match with B. B is going to match with A. So, if it is being treated with both, now antigen and antibody matches, now that is going to be the matching point. Now, I start with mixing the RBC of a person, and that is being mixed with anti-A antibody serum, which contains B. If clump is formed, the blood group is assigned as A. If that particular person's blood is B group, now RBC is of B group, treated with anti-serum B antibody containing A antibody, then if it is clump is formed, the group is assigned as B. If clump is formed both with anti-serum A and anti-serum B, then the group is designated as AB. If no clump is formed either with anti-serum A nor B, then the group is designated as O. This is how the antibodies are being used to test in blood typing. Break. Okay. Now, let us start with the genotypes. This is the genotype for A type of blood. And this is the genotype. This is the genotype for B type of blood. And this is the genotype for AB type. And this is the genotype for O type of blood. Now altogether there are four phenotypes, 6 genotypes. Now this is the one which contains the antigen A, this is the one which contains B, this contains AB, this does not contain neither A nor B. Coming to the antibody, this is antigen. Now antibody A group, it will have B, and for B group it has A. For this. Now they are the ones which are having neither A nor B. Now these are the ones which are having both antibodies A and B for O group. So antigen and antibody. Now here once we talk about what type of blood they can tolerate, to whom it can give. Now what I can give is, this is the one, this is the one. What type of blood it can tolerate, to whom it can be given. Now A type of blood can be given to A blood, and that is the one which can also be tolerated with O. And this B group, they can also tolerate the O. And this AB, they can tolerate AB. And this is the one which is going to have the toleration only with the O group. Now, to whom it can be given? They can be given to A or AB. Now, they can be given to B or AB. They can be given only to AB. Now, O type of blood can be given to O. It can be given to A. It can be given to B. It can be also given to AB. Now you can see of all these, O type of blood is the one which can be given to altogether all the other three groups apart from its own group, and AB group is the one which can receive the blood from A group, B group, AB group as well as with O group. Hence it is the one which is being received as called as universal recipient, and O is the universal donor for any group. Break. A Delhi public school student who is a middle class student, he explained the easiest way to understand the blood transfusion. What he has demonstrated in a science exhibition is just he has taken 4 types of beakers. This is beaker number 1. He has filled it with water. Now he has taken another beaker. He has also added that particular water, and for this he added a chemical which gives a red color. Now this he called it as O type of blood. This he called it as beaker number 2 as A group of blood. Similarly, he has taken the third beaker. Now he has added that particular water. To that he added a chemical which gives blue color, complete blue color, dark blue color. This he called it as B group. This is colorless, this is red, this is blue. He has taken a fourth beaker and he filled with water, colorless, and now to this beaker he added the chemical that is added to the second beaker, and also the chemical added in the third beaker. Now it has both red as well as the dark blue color. Now this is what he is referred as AB group. Now this is colorless, this is red in color, this is blue in color, this is the one which is having both red as well as blue. Now this first beaker, it can give blood to, can mix with second beaker, It can also mix with third beaker. It can also mix with the fourth beaker. Because it is the one which can easily mix. That means O type of blood can be given to anybody. It can mix with its own. It can mix with A. It can mix with B. It can mix with A and B. A group of blood can be given only to A as well as to the fourth beaker, that is AB. A type of blood can be given to A or to AB. Similarly, the third beaker can mix with either to its own or it can be given to the fourth beaker. B type of blood can be given to B or to AB. That is why the terms universal donor and universal recipient. In blood transfusion, the antigens is important for the donor. The antibodies are not significant. For the recipient, antibodies are important and antigens are not taken into consideration at all. Now, antigen, what it will do is, it is going to have reaction with that particular antibody. If the antibody matches with that particular recipient, then there is no question of agglutination. If that antibody matches, then there is the process of agglutination. Now, this particular O antigen, O group, has neither A nor B antigen. That means it will not having any specific interaction with that particular antibodies present. Hence, the O is called as universal donor. Now, AB group, it is not having neither A antibody nor B antibody. That means it is not going to produce any antagonistic reaction, any action opposite. That is why it is referred as universal recipient. Universal donor and universal recipient. The whole concept of this particular inheritance is due to that particular gene present on the 9th chromosome, as an example with reference to the multiple allelic forms of that particular gene. So in the next, I am going to talk about another concept of the blood. Every blood, either it may be either positive, A positive or A negative, B positive or B negative, AB positive or AB negative, and O positive or O negative. This concept of positivity, negativity can be understood when once we discuss about erythroblastosis fetalis, which is none other than the RH system, which is again expressed by the multiple systems, and these multiple alleles are the ones which are going to cause further this particular 8 types of blood groups with reference to the phenotypes, and genetically they are different.